All right, thank you very much for your interest in my presentation here at DigiCrop. So the full text of the full length presentation will focus a lot on how we can prioritize genes with phenotypic impact in the genome, which will benefit both plant genetics and plant breeding. In this shorter version of the talk, I'm going to jump straight to the conclusions. So one real interesting thing about the genome is that if you look at those genes that are actually linked to loss of function phenotypes in, say, maize, they are very different from the average gene model in the genome. They are much more likely to have evidence of actually being translated into protein. They tend to be expressed across more tissues. They tend to be synthetically conserved, so in the same place in the genome of maize and also related species like sorghum and foxtail millet. They're also just longer and have more exons. One thing that really distinguishes a lot of the gene models in the genome which are not linked to phenotypic variation is they're more likely to be involved in presence-absence variation, which is the sequence of the gene model is present in some varieties of maize, but absent in others. One of the questions I get when I show this result is this some sort of marker of essentiality or how severe the phenotype will be from a loss of function mutant? It's a very good question. In maize, I can't answer it because we only have about 100 of these genes to work with. But in Arabidopsis, we've been fortunate to benefit from the work of hundreds, if not thousands, of researchers who have cloned and characterized a larger number of genes, and we can divide the phenotypes of these genes, the loss of function phenotypes of these genes, into distinct classes. And what we see, this axis is summarizing a lot of the same features I showed you in the previous slide, is that whether a gene produces a lethal phenotype, a constitutive, a non-lethal phenotype, or a conditional phenotype, we essentially see this same distribution of the feature. So this doesn't really seem to be a marker of essentiality. It's just, is there a phenotype from the loss of function mutant or not? The next question is, okay, so this is loss of function qualitative phenotypes. Does the same pattern hold up for quantitative data? And it does. So what you see here is data from both conventional GWAS and GPWAS, genome-wide phenome-wide association studies, something I'll tell you more about in the 15-minute version of my talk. And we see the same pattern. So genes linked to quantitative genetic variation, they're more likely to be expressed, they're more likely to be synthetically conserved, they're less likely to exhibit presence-absence variation. They also seem to be under stronger purifying selection, like classical mutants with loss of function phenotypes, and unlike the average gene model in the genome. And in fact, if we overlay classical mutants and genes identified through a quantitative genetics method, in this case GPWAS, they show essentially the same distribution on this axis, which is very different from the remaining gene models in the genome. This is very exciting to me because I think what it allows us to do is to start predicting which gene models in the genome which are not yet linked to phenotype actually have the potential to produce phenotypes when disrupted, and which ones are unlikely to. I saw a lot of my colleagues in grad school assigned projects where you order up some loss of function alleles of a mutant in Arabidopsis or in maize. You spend several years looking for a phenotype, comparing the mutants to the wild types, and you don't find anything. So through this approach, I think we can rebalance the risk-reward of doing reverse genetics and incentivize people to start characterizing the function of the vast majority of plant genes about which we know nothing. Thank you very much for your interest. I will be happy to answer questions, and I hope you will be interested in seeing the longer version of my talk.